Hello, it's me, Oscar from Marijuana Garden.
Based on the image content and current scientific knowledge, the claim, "This is the most detailed view of a human cell to date," is, misleading and likely inaccurate. Here's a detailed breakdown. First, the visual is clearly a three-dimensional artistic illustration, not a scientific micrograph or, an actual photo or microscope image. It uses dramatic lighting, vibrant colors, and those perfectly smooth textures that are, honestly, typical of computer graphics. Real, high-resolution microscopy images, like those from Cryo-EM or super-resolution microscopy, have distinct characteristics, such as grayscale or limited color, which is often added artificially later, as well as noise or grain, and specific labeling for structures. Second, most detailed is kind of unverifiable and, exaggerated. Detail is actually pretty subjective. While the illustration shows many organelles at once, actual scientific images achieve far higher resolution for specific structures or molecules. Techniques like Cryo-Electron Tomography or Cryo-ET can resolve structures down to near atomic levels, in angstroms, within a cell, providing vastly more molecular detail than this illustration depicts. Projects like the Allen Cell Explorer create highly detailed predictive three-dimensional models based on vast data. But they are still labeled as models or visualizations. Third, there's a lack of specificity. The post doesn't state how this view was obtained, who produced it, or what data it's based on. Genuine scientific breakthroughs are always published with methodology and source attribution. So, what does the image likely represent? It's probably a high-quality scientific illustration or visualization created by a biomedical artist or a scientific visualization studio. It's a composite representation that combines knowledge of many different cell structures, like the nucleus, mitochondria, Golgi, cytoskeleton, vesicles, and so on, into a single, aesthetically pleasing image. Its purpose is educational or conceptual, really, to give viewers a sense of the incredible complexity and organization within a human cell. And honestly, it succeeds brilliantly at this. So, here's the thing. Science doesn't actually have one single image that shows everything inside a cell at maximum detail, all at once. Different imaging techniques are each good at capturing different scales and components. For instance, light microscopy lets us see whole living cells, but its resolution is limited to about 200 nanometers. Then there's super-resolution microscopy, like STED or PALM and STORM, which can actually beat the diffraction limit of light and resolve structures down to about 10 to 20 nanometers. Electron microscopy, or EM, goes even further, providing nanometer-scale resolution. And honestly, Cryo-Electron Tomography, or Cryo-ET, is especially powerful here. It lets us image cells in a near-native state at high resolution, revealing what some scientists call the molecular landscape, or even molecular sociology. If we want to see the atomic structures of isolated proteins or molecules, we use techniques like X-ray crystallography or Cryo-EM single-particle analysis. But remember, these only work on isolated pieces, not the whole cell at once. Most advanced microscopy techniques, by the way, actually produce images in grayscale. Color is usually added later, computationally, to highlight specific structures or molecules, what we call false color. Now, about the truthfulness of the image in question. The phrasing really does imply that it's a direct scientific image showing the ultimate level of detail, but that's just not the case. If you interpret it loosely, as a conceptual illustration that shows the complexity of a cell, then sure, it's valuable and impressively detailed from an artistic or composite perspective. It does represent visually what we know exists inside a cell. If you're interested in seeing actual detailed views, there are some fantastic resources out there. First, Cryo-Electron Tomography, or Cryo-ET, is really at the frontier for imaging intact cells at high resolution. You can check out the Cell Image Library online. Just search for Cryo-ET datasets there. For example, look for research from labs like Wolfgang Baumeister at Max Planck, Grant Jensen at Caltech, or Peijun Zhang at Oxford. Their studies often focus on imaging things like mitochondria, the nuclear pore complex, or synaptic vesicles inside cells. Another great resource is the Allen Institute for Cell Science, especially their Allen Cell Explorer. They create integrated, predictive, three-dimensional models of human cells based on huge amounts of imaging data using fluorescence microscopy. It's important to note that these are models, but they're grounded in real data. And just to put this all in context, the Nobel Prize in Chemistry was awarded in 2014 for the development of super-resolved fluorescence microscopy. and again in 2017 for Cryo-ET. These are really the key technologies behind our current most detailed scientific views. So in short, the image we've been talking about is a beautiful and informative illustration, but it's definitely not the most detailed view of a human cell to date in the scientific sense. The real cutting-edge views come from techniques like Cryo-ET, which give us unprecedented molecular detail within the cellular environment, but not as a single whole-cell colorized picture like this one. It's always a good idea to be cautious about science claims you see on social media, especially when they don't include specific sources or details about the methodology.
Thank you.